Hi, everyone. Thank you for joining the 7th webinar in our series on the use of NAMs in risk assessment. This series is being co-organized by the EPA, the Physicians Committee for Responsible Medicine, and the PETA International Science Consortium. My name is Amy Klippinger. I direct the PETA International Science Consortium, and I'll be moderating today's webinar. We have one speaker today, Dr. Steve Simmons, who will be presenting on identification of NAMs for placement on the TSCA Section 4H2C list, a proposed NAM nomination form.
After the presentation, we will have 4 panelists representing different sectors responding. Before I introduce Steve, I'd like to point out that slides from today's webinar and a recording will be posted at the link on the screen shortly after the webinar. Slides and a recording of the first 6 webinars of this series are also available at this link. There will be time for questions and feedback for our speakers at the end of the panelists' presentations. Everyone is on mute. You can type any questions in the questions section within your GoToWebinar toolbar, which should show up on the right-hand side of your screen. And with that, I'll turn it over to Dr. Gino Scarano for a second to give a brief introduction as well.
Thank you, Amy, and good morning, everybody. I'm excited to have Dr. Simmons present this proposal and hear feedback from the stakeholders that have agreed to come and provide some feedback. As many of you know, we've been working hard at EPA in trying to best work through Section 4H of the amended TSCA, which calls for the reduction in the use of testing in vertebrate animals. And this proposal is part of meeting that mandate in Section 4H. And we view the presenting of this kind of a proposal and getting feedback as an important part of collaborating with our stakeholders and hearing from people who are listening as to how we can best do this work. So thank you, Amy, and good luck, Steve and panelists, and I'll turn it back to you, Amy.
Great, thank you, Gino. So speaking first today, as we said, will be Steve Simmons. Steve has been a toxicologist with the U.S. Environmental Protection Agency since 2006. While at EPA, he has worked in the Office of Research and Development, beginning with the National Health and Environmental Effects Research Laboratory, later with the National Center for Computational Toxicology, and currently serves in the Center for Computational Toxicology and Exposure. In his 14 years at EPA, he has developed various high-throughput screening assays and has developed cost-effective solutions to retrofit high-throughput assays to incorporate metabolism. Steve?
Thank you, Amy. I want to thank everybody and the organizers for giving us the opportunity to present this material. This is a project that Gino and I started working on late last summer, and I just want to say up front, it's a small part of a much larger effort to incorporate new approach methodologies into TSCA. Gino, sorry, hang on just a second. So Gino already sort of alluded a little bit of this, but we'll lay out a little bit of the legal background as to why we're doing this. The Toxic Substances Control Act was amended in the summer of 2016 by the Frank Lautenberg Chemical for the Safety for the 21st Century Act. And in that new law, it included a subsection that requires EPA to promote the development and implementation of alternative methods and strategies to reduce and refine animal testing, under the proviso, though, that it gives us information of equivalent or better scientific quality and relevance for assessing risk to human health and the environment. And so that subsection 4H has 3 main parts. The first part is on this slide. I'm not going to read all the bullets in the interest of time, but this first part of section 4H describes what the EPA has to consider before it can require animal testing. Section 2 under 4H has 6 parts. We're only going to focus on one today, and that is the requirement that EPA post a list of alternative test methods or strategies, what we're referring to now as new approach methods or NAMs. And One of the things the law calls for is that the EPA publish a list of NAMs that it's currently using, which we did in 2018, and that we update that list annually. And so the first update to that list was released in December of 2019. But additionally, we have to release a draft proposal on a process for selecting new stringent guidelines so that it ensures that you get high quality NAMs. There might be other NAMs that might be useful for TSCA decisions. So we would like to propose some ideas on how NAMs could be considered for inclusion on the existing TSCA list. So in order to replace animal testing in the future, one of the assumptions we work from is that we're going to need many high quality NAMs that we currently do not have. So the immediate goal of this process should be to solicit, find, and develop as many of these NAMs as possible. And unless we at the EPA want to find and develop these NAMs ourselves, we need to encourage outside interest to nominate NAMs, or at the very least, we need to be cognizant that we don't do anything in this process that discourages submissions of NAMs for consideration. We need to provide guidance to the outside community as to what we do under TSCA in terms of the types of risk decisions we make, how NAMs with various endpoints might help with that in terms of the context relationship between the endpoint and the risk decision. And then we also need to provide descriptions of what sort of metrics we would use to evaluate NAMs for use in terms of acceptance criteria. And so I want to go ahead and give a spoiler that I'm not laying out any acceptance criteria today. That's really beyond the scope of what I worked on with Gino over the summer. I mean, I think it's perfectly valid discussion point for later, but I'm not going to be talking about evaluation criteria today. So those are the practical goals. Ideally, what would we like for a NAM to look like? Well, in accordance with Administrator Wheeler's memo from September 2019, we would like a method that does not rely upon intact animals. The TSCA refers to vertebrates specifically. Andrew Wheeler's memo uses the word animals. So, you know, there's some ambiguity in exactly not in the law, but in terms of policy, maybe whether or not we're trying to reduce animals specifically or vertebrates specifically. And I would say trying to reduce our reliance on intact animals, period, is probably the ultimate goal. We would, another key feature would be that it be relevant, which is a term I'm going to attempt to define a little bit later, for the assessment of new and existing chemicals. This would include the 86,000 chemicals currently on the TSCA inventory, as well as new chemicals that are coming to into TSCA workflows. Ideally, a NAM would provide quantitative data rather than simply a qualitative plus minus outcome, so that thresholds that we would use from that NAM for risk decisions could be revisited and adjusted if we need to. Another thing to consider is the TSCA chemical space. I mentioned there's 86,000 chemicals and, you know, roughly 2,000 being added every year. It's a vast and diverse chemical universe with everything from small volatile molecules to large, rather undefined polymers, with wide ranges of phys-chem properties like log p-values, solubilities. There's diverse use classes. There's mixtures, defined and undefined, and so it's sort of an unruly chemical space, not just with respect to the number of chemicals, but the types of chemicals that are on that list. So we would like NAMs that we're able to test as much of that as possible. NAMs would need to predict known values where there are good positive and negative control data. NAMs should be highly reproducible, not only within a single laboratory, but across laboratories using a standardized protocol. NAMs would ideally be accessible and transferable. And by that, I mean that they use techniques, equipment, and reagents that are common or easy to acquire. And lastly, NAMs should be as transparent as practical, meaning that they should avoid reliance upon proprietary methods, data streams, or algorithms. But we realize going into this, there's going to be commercial entities that probably want to nominate NAMs. So it's not a, obviously these aren't hard requirements. This is just ideally what we'd like things to look like. So now that we know what an ideal NAM looks like, the question is what types of NAMs or NAM nominations are we likely to get? And the answer is less than ideal, right? So depicted by the picture here on the right. That's okay. A perfect NAM doesn't exist. It's probably never going to exist. But I think we should clearly articulate what the ideals and optimums are for a NAM, and so that people who develop or look for NAMs know what we're aspiring to. So the first step then is to establish a portal for NAM nominations. A perfect NAM would not rely upon intact animals. It would provide relevant data, which I'll try to define the term relevant here in a few seconds. That covers risk assessment activities for new and existing chemicals. That provides quantitative rather than qualitative kind of data. It covers the diversity of TSCA chemical space, highly predictive of known values, that it's reproducible both within the same lab and across laboratories using a standardized protocol, that it be transferable and accessible, and that to the extent possible that it be transparent. So I'm going to go to slide 8.
Okay.
And can everybody see slide 8?
Yep, I can see it.
Okay, great.
Beautiful picture. Yeah.
Okay, great. So I mentioned that, we've set out the ideal, what are we likely to get? Probably something less than ideal. I mean, actually, almost certainly something less than ideal. That's okay. Perfect NAM doesn't exist, so we're going to be dealing with a spectrum of imperfect NAM nominations. They all are. But we need to clearly identify what it is we're looking for, so that as people are trying to either develop, repurpose, or find NAMs suitable for TSCA use, they know exactly sort of what the goalposts are. All right, I'm going to advance to slide 9. So the first step we have to do is establish a portal for NAM nomination. And I want to reiterate that NAM nomination is not the same as evaluation. In this cartoon, we are the door, not the person sitting behind the desk. The aim of the nomination process would be, the aim of the nomination process should be to allow the TSCA NAM team to triage the nominations we do get into those that might be turnkey ready for TSCA use, those which clearly miss the mark and probably will never be suitable for TSCA use, and then the third category would be those depicted here in the cartoon, which show a little bit of promise but might require some additional information or development before we can make a decision. So now I'm going to advance to slide 10. So who's the target audience for this nomination form? Well, we can envision several groups who might want to nominate NAMs, and each of these diverse actors offer unique advantages. Each will probably have their own reasons for nominating a NAM, which is something I'd like to explore later in the Q&A with the panelists. Some groups will be very familiar with TSCA, while others less so. And so some might be better at certain NAM types than others. And these things might color their assumptions about how a NAM would be used. So I'm going to slide 11. So my strategy was to use the things that are available, which was open EPA source material, guidance documents from places like OECD, ICVAM, and ECVAM, and ask what kind of NAM inputs would mesh well with TSCA. There are several NAM types, which we'll talk about in a few slides. What information did these source documents request for the various different NAM types? How much overlap was there in the requested info? Was there a way to harmonize all that information? Also, remember, we're trying to induce people to submit. So we want to be careful not to set the initial bar so high or place an undue burden with respect to the volume of the information that we're requesting. If a NAM relies on proprietary data or methods, one of the questions we have to grapple with, which I don't know if we have an answer to yet, is whether or not we can protect private business information. I'm hoping that we can, so that people who have commercial products that might be good for TSCA would be induced to submit. But I've distilled the original list of 8 criteria from the EPA strategic plan on NAM use for TSCA down to 5 critical elements that capture what I think we need for NAM nominations. And these are the 5 basic elements that I then use to structure the proposed nomination. And those are nominal information, developmental history, method description, relevance, and reliability. So I'm going to slide 12. So the nominal information, I'm not going to spend any time on this, is just that we just need the name, the nominating official, what type of NAM it is. When I said there's many types of NAMs, these are the types of NAMs that we anticipate getting, although I should mention it's not necessarily an exhaustive list of NAMs. There could be other NAMs that we just don't envision yet. Section 4 of the EPA strategic plan suggests identification and development of NAMs for chem characterization, hazard, that being human health and ecotox, as well as dosimetry and exposure as separate from the integration of NAM. That's why I have the integrative method highlighted in red. However, if you look at the Wheeler memo, it specifically defined NAMs there as any technology, this is a quote, any technologies, methodologies, approaches, or combinations thereof that can be used to provide information on chemical hazards and potential human exposure, end quote. So I've included integrated methods here as a NAM type. So I'm going to the next slide, 13. We asked for the developmental history, and this is in the case that the nominator is not the developer. We may need to it might be more appropriate to contact the developer for clarification or additional information rather than the person who nominates. Next slide. Method description is pretty straightforward. We're asking for a description of the method, or protocol, and steps, so we can sort of understand what's going on with the method. Tell us what the endpoint is, how is it measured, modeled, or predicted, what types of values are outputted from the method, describe any calculation methods that are used. If it's a model, can you please tell us what features or descriptor sets you built the model upon? And then we ask that you give us some sort of rough outline of the throughput and resource intensity for the current version of the NAM. So for instance, if this is an in vitro method, is it a 384 well plate type assay, and how many plates can a single person in a single day run at what kind of costs, that sort of thing. Next slide. Okay, so relevance is one of those things that we talk about a lot, but it's not very well defined. So in the strategic plan, we use the OECD definition from their guidance document 2005, and that is that relevance is the ability of a test method to measure or predict an effect / target of interest, as well as the regulatory need, usefulness of the alternative methods, and associated limitations of the test method. And in the strategic plan, there were 2 criteria that dealt specifically with relevance. I'm not going to read those, but effectively, the way we envision relevance is it's splitting into 3 sub-elements, and that is decision context, the relationship of the NAM endpoint to that context, and chemical coverage. So next slide, 17. So in the decision context, we're asking for the nominator to tell us which risk decision context they envision the NAM endpoint being useful for, whether that's pre-prioritization, prioritization, or risk evaluation, which are 3 activities that are typically associated with existing chemicals, and then screening level assessments, which is a new chemical specific activity. Next slide. The endpoint to context, the NAM endpoint to risk decision context is something where we envision providing a checkbox. And so this is a distillation of endpoint and effects that I've got from ChemView, which is our repository for information on chemical health and safety data received by EPA's assessments and regulatory actions for specific chemicals under TSCA. So it seemed appropriate to use here, at least as a starting off point. And so each of these five endpoint to context relationships will be subpopulated with a series of additional sub checkboxes that you can use. So for instance, for under hazard ecotoxicology, the next to the last one here on this list, we would have acute aquatic plant toxicity, chronic terrestrial plant toxicity, and then acute and chronic aquatic toxicity for algae, invertebrates, and fish. So if your method deals with, obviously fish, then you would check that box. All right, next slide. So we ask that you provide a brief description with supporting references for the rationale linking the endpoint to the decision context. If it's a biological sort of endpoint, does it map to an AOP? And if so, which AOP? That's the adverse outcome pathway. And then we ask whether or not the NAM provides qualitative versus quantitative data. Next slide. So the last sub-element under relevance is chemical coverage. We asked for you to describe the method used to define a chemical applicability domain and to describe any other chemical limitations to the NAM, such as DMSO solubility, vapor pressure, et cetera. And then chemicals known to produce false positive and negatives. I think asking for a chemical applicability domain makes sense for certain NAM types, obviously things like QSAR models or PhysChem models. Those are going to be trained on hundreds, probably more likely thousands of chemicals. For other NAM types, like say an in vitro assay, I think the best we can hope for is this, a substantive answer to the second question, which is known chemical limitations, simply because they're typically trained on fewer chemicals. And even for the first question for QSAR models, there's really no set way to do this. If you're familiar with literature in the QSAR community, people use different predictors and features. They use different methods for defining distance metrics first, and then there's people who prefer novelty detection versus distance to classifier boundaries, et cetera. So it's difficult. Even in QSAR language, it's difficult to describe bounded space in easily understandable terms. So we're asking you to, if you've done either one of or both of these things, to basically give us some idea, at least with respect to the chemicals that you have tested, how that sort of pairs up with the TSCA chemical landscape. Next slide, 21. So the last critical element is reliability. And again, we use the OECD definition in the strategic plan, the definition being the extent of reproducibility of results from within and among laboratories over time when performed using the same standardized protocol. And 6 of the 8 criteria in the strategic plan dealt with reliability. I'm not going to read them. Next slide. So it should be on 22. So the 4 sub-elements of reliability, as they were distilled down, are reference chemicals, reproducibility, predictivity, and transparency. And so for reference chemicals, here we're looking for 2 things: the controls and standards used in every experiment to qualify acceptance. And then the second thing we're looking for is the training set of known positives or negatives used to evaluate whether or not the assay is predictive of known values. So that's the purpose behind the 2 questions listed on slide 23. Next slide. So for reproducibility, what we're looking for here is how reliable the assay is from experiment to experiment within the same lab. What are the QA criteria used to establish whether or not an experiment is accepted or failed, and any limits of detection or quantification. Next slide. So for predictivity, here what we want to know is how the NAM was evaluated with a training set of chemicals, as well as any known blind spots related to assay artifacts, assay interference, false negatives, false positives, etc. Next slide. So transparency. This sub-element gets at both transferability and transparency, asking if the method's been used successfully beyond the lab in which it was originally developed. And then also if any sort of intellectual property issue, are there any sort of intellectual property issues that might prevent an outside evaluation, a thorough evaluation. So we ask if it's been evaluated by outside users. If so, can you give us a list, describe the sort of reproducibility using reference and control chemicals, any sort of independent reviews that may have been performed. So this would include, if it's currently under review with OECD or ECBAM, et cetera. And then describe any sort of specialized proprietary equipment, software, or data that might impede NAM transfer to a third-party user so that it could be independently evaluated outside of the laboratory of the developer. So next slide, 27. So this is not an exhaustive list of unresolved questions. This were just 3 that I had sort of jotted in the margins as I was working on this. One of the main things that I think Gino and I would like to get out of this to help us sort of refine this nomination process is to learn from people who might nominate a NAM is what would be the sort of motivations or incentives you would need to submit a NAM for use under TSCA. Because although I came at this process, Gino and I came at this process with that sort of in mind, that we want to encourage people, incentivize this. I'm not sure as somebody who works inside the agency that I have a full picture of what sort of incentives might drive somebody outside the agency to nominate a NAM for TSCA use. Second, this gets at the question of, I mentioned earlier with NAM types about integrated methods versus individual methods. If we do include integrated methods as one of the NAM types that we would consider for use under TSCA, is it necessary that we evaluate all of the constituent methods that go into an integrated approach independently, or can we just evaluate and adopt an integrated approach comprised of unevaluated components as long as we're evaluating the output of the integrated method itself? And then lastly, can we include phys-chem methods as NAMs? And the reason I asked that is this came up in an internal deliberation about this process. There was a thought, and if you'd asked me a year ago, I would have agreed with this, that phys-chem properties, we don't test animals for phys-chem properties, so a method that predicts a phys-chem property wouldn't really be a NAM because it's not really replacing an animal test. But as part of the project of working with Gino, I actually got to go to DC and sit through actual TSCA cases and watch them, watch decisions actually being made on not existing, but on new chemical cases. And I knew this sort of cognitively, but it was remarkable to sort of see in person how much phys-chem properties are actually used every single day to make decisions about manufacturing, import, requests for new tests, et cetera. And so I thought, I've changed my mind. I think it might actually be important to have phys-chem methods as a NAM that we would consider including. I mean, so if you look at some of the NAMs that are already on the list, some of those predict phys-chem properties. But I guess the real question to pose is, do NAMs necessarily need to replace an animal test to reduce animal testing? So those are just some ones I thought I would throw out, but I'm really anxious to hear what not only the panelists have to say, but the other folks who've registered online and might ask questions through the Q&A. So that's the end of my slide presentation. So with that, I guess I'll turn it back over to Amy.
Great, thank you, Steve. So that was a great presentation. I'm looking forward to having some Q&A on it at the very end. But before we get to questions and comments from our audience, we're going to have 4 panelists speaking, each for 5 minutes. First up is Lauren Heine, who is the Director of Safer Materials and Data Integrity at ChemFORWARD, where she focuses on green chemistry, green engineering, and multi-stakeholder collaboration to develop products and processes. Lauren led the creation of the web-based platforms Green Screen for safer chemicals, the Green Screen List Translator, and Clean Ingredients. So Lauren, we'll turn it over to you now. We have your slides up on the screen.
Thank you very much, Amy. Can you hear me okay?
Yes, we can.
Great, thank you. And thank you to Gino and Steve for this presentation and for opening this up to the public for input. I think this is really important work. Next slide, please. I'd like to just give you a little context for my organization and our interest in NAMs. ChemFORWARD is a nonprofit organization committed to becoming a trusted source of cost-effective, curated chemical hazard information on safer alternatives. And we believe that increasing the use of comprehensive chemical hazard profiles will lead to proactive decision-making to help users eliminate chemicals with high hazards, avoid regrettable substitutions, find or substantiate the availability of safer alternatives. This can help with local regulations, facilitate transparency in the supply chain, design safer products from the beginning, and ensure a safer, circular, and more equitable economy. Next slide. We work with chemical manufacturers, retailers, brands, and pretty much the entire value chain. By chemical hazard assessment, we're referring to the systematic process of assessing and classifying hazards and phys-chem properties across a spectrum of endpoints and severity. Most chemical hazard or CHA methods are based primarily in the globally harmonized system of classification and labeling, but they also go beyond it. There are probably the most well-known methods are the cradle-to-cradle material health method, the EPA safer choice AA criteria method, and the green screen for safer chemicals. And these, as I mentioned, these methods use DHS but build on them, adding things like endpoints for endocrine disruption or persistence or climatic relevance, things like that. But they go as far as they can with GHS and then beyond. Next slide, please. So how the program works. Oops, there's one slide missing. Can you go back? Thank you. Basically, what happens is if a safer, well, first of all, if there's a chemical of concern, we look for a suite of potentially safer alternatives that have been demonstrated to be used and are potentially functional options. And so we identify these chemicals and then hire a licensed toxicology profiler to do the assessment. The assessor enters into the back end of the ChemFORWARD application their entire data summary, and this could include traditional toxicology data and NAMs. There are already a number of NAMs used in chemical hazard assessment, typically EpiSuite and Oncologic and things like that. We would find it very valuable to have NAMs for phys-chem properties because sometimes there are data gaps for that. So the assessors assign hazard classification levels. They enter their level of confidence. They prepare a rationale for why they classified the hazard for that chemical the way they did. And then they submit the CHA for what we call verification. So, as the assessor is a professional toxicology firm, the verifier is an individual expert in toxicology who it acts as a peer reviewer. They judge the CHA for completeness, comprehensiveness, consistency, quality. They also need to judge the appropriate use of NAMs. And so within the app, there's a commenting function that happens. The verifier makes a comment, the assessors respond, they go back and forth until all the issues are resolved, and then the verifier flags the CHA as verified. And that CHA is posted to what we call the front end or the user side of the application, where you can see the chemical properties, you can see some manufacturer information, you can see the same data entered into the backend presented in two different summary hazard tables. You can see it presented using GHS criteria only, and you can see it presented using cradle-to-cradle criteria only. And more views could be added, but again, same data on the back end, different views on the front end. And these assessments are subject to continual improvement. We have a program for accepting technical challenges, and we're always challenged by filling data gaps. The value of these assessments is it tells you what's known and what's not known about chemical hazards based on the best available information. And ideally, going forward, we'll fill data gaps more and more. Next slide. So from our perspective, NAMs are essential. We're excited about what EPA is doing, and the questions or challenges I'm going to raise are going to be good problems to have once EPA has brought more and more NAMs into the fold. But training will be absolutely essential. So there will be more and more NAMs, and organizations like ours who hire professional toxicology firms and professional toxicologist consultants will need to know how to use all of these different NAMs, and how do we ensure that the different assessors know how to use the same NAMs, or will they use different NAMs because some are more expensive than others? So some firms may have access to certain models and others don't. So I can imagine challenges around really accessibility to these NAMs. Are they affordable? What can EPA do to ensure that enough training is available? How about starting in the universities? How about for professional education? How, as an organization, can we ensure that our assessors are using NAMs consistently, appropriately, that kind of thing? And for NAMs are going to be extremely useful in applications like ours, which are not regulatory applications. How challenging will it be to translate the diverse information that comes from NAMs into recognizable hazard classifications like GHS, the categories, things like that, and into classifications for other types of programs, for example, certification programs. So we're really sort of practical, on the ground, help people who make products make better decisions. And so we want to use these NAMs too because they're filling critical data gaps. And then finally, as a citizen, I think it's important to have enough expertise to really be able to judge the quality of the hazard assessments produced by others. And using NAMs, will there be enough transparency and expertise outside of EPA to have enough intelligent users and consumers of these NAMs to judge their use? So I think you can see my perspective here, and excited about where you're going with this, and excited to implement it as soon as possible in a way that will support greener chemistry and engineering going forward. Thank you.
Thanks very much, Lauren. That was great to get your perspective. And Steve, after the four presenters or the four panelists, I'll give you a chance to respond if you'd like. But for now, I will introduce our next panelist. Next is Patrick McMullen, who leads the Computational Toxicology Group at ChitoVation, which is a contract research organization focused on pioneering cell-based and computational methods using innovative science to develop safer and more effective pharmaceuticals, food ingredients, agricultural chemicals, commodity chemicals, and consumer products.
Perfect. Thank you, Amy. I see my slides. Thanks, Amy, for the introduction and for putting this together, and thanks to Gino and Steve for undertaking this. What I'm sure has been a challenging intersection of statutory requirements, science, and public health. So happy to be a part of the process and give a little bit of feedback and commentary on what you've presented. ChitoVation, for those of you who don't know us, we're developers of NAMs. We develop NAMs and we work with stakeholders being companies, nonprofits, and government groups on implementing those NAMs to make responsible safety decisions. So I lead the Computational Toxicology Group. That probably flavors my comments a little bit. We do a lot of research and development, and I've been thinking about what Steve just presented in terms of kind of how our company would approach taking some of the technologies that we use and that we're developing and applying them, applying for inclusion on this list. So a couple of points I wanted to raise. Most of them have already been touched on. The first is that there's an incredible diversity in the way NAMs are used, even within a single TSCA risk decision context, right? There's differences in the amount of information that needs to be generated for compounds that, for instance, have a large data gap versus compounds where there's maybe are already in use. And there's a lot of a lot of data on that compound or on its chemical neighbors. Similarly, in a risk-based approach, a large margin of exposure may indicate a lower standard of evidence might be required for the NAMs that are used to characterize safety. So I want to come back to, and I'll come back to this at the end, Steve's last point about replacing animal tests. When in most TSCA decisions, there isn't a single animal test that's being considered. So we need to be thinking about, from more of a weight of evidence perspective, how these are going to be used and what that means in terms of how much validation, how much characterization of relevance and reliability is needed. And from Steve's presentation, I think that's, there's an acknowledgement of that. And I encourage that to continue as this continues to develop. I think I'm sure we're already leveraging existing guidance programs. I look at the list. The list is mostly OECD guidelines. Amy pasted that in the chat just a couple minutes ago. You can take a look. Now those are very, very well validated, very well accepted processes. And I guess my question and what I think we need to work on clarifying a little bit is what's different about the assays and the goals of the TSCA program for including assays and including NAMs on this list. What's the difference between what's being expected from OECD, for instance, test guidelines and this process? I think there's a lot of lessons we can learn from ICVAM, ECVAM, and also programs like looking at like the FDA biomarker qualification plan, which different context, but similar, a lot of similar concepts in that we're developing a surrogate for some kind of endpoint. Briefly, considerations for relevance and reliability. We work in a group and we work on a diversity of applications. Some of them are very simple cell-based assays targeting things like cytotoxicity and things like that. And others are very complicated organotypic culture systems that maybe have a complicated exposure workflow, such as we're developing a respiratory model with the Reproducible system. And Steve showed this plot of characterizing the chemical space that had hundreds of dots on it. And he mentioned that the reality is that for a lot of these systems, we're lucky to have maybe 5 or 10 compounds tested. And is that my question is that a showstopper? And is how much can we rely on the scientific credibility and our understanding of the similarities between the biology that's evinced in a NAM and the biology that's in the intact organism as a kind of to stake our validation on. With regards to what would motivate us to submit a NAM, I think that a challenge of TSCA in general, and I think we're seeing it here, is to establish some clarity on the process so that submitters know what to expect. This is nomination for inclusion on the list. These nominations get passed to a committee that's going to evaluate them, but for someone like me, not knowing what that evaluation is going to entail sort of dissuades me from spending the energy to nominate in the first place. So, and understanding where we're setting the bar relative to other programs, I think is an important consideration as well. So I think that's my time. Thanks again to PETA, PCRM, and EPA for putting this together, and happy to discuss any of these topics further.
Thanks so much, Patrick. That was really useful feedback. And just a quick reminder again to our listeners that we will have time for questions to Steve after our 4 panelists. So if you have questions or comments, you can type them in that question section in your GoToWebinar toolbar. Our third panelist is Pam Spencer. Pam is the Vice President of Regulatory, Product Stewardship, and Quality at ANGUS Chemical Company. Prior to this role, Pam worked for the Dow Chemical Company for 30 years. Her work advances safer product design and the application of NAM.
Thanks, Amy. Can you hear me okay?
Yes.
Perfect. Let's go to the next slide. And I just want to thank Steve and Gino for that presentation. It was very helpful to kind of see what the process is to get in the door, and I appreciate that. And I think that will really help to serve as a frame of reference as we're thinking about how we use these NAMs, at least in the private sector. So we heard from Lauren on uses beyond, EPA, and Patrick from a developer. So I want to speak a little bit to a user and try to answer hopefully some of your questions. And I really want to frame this in the context of new product development and not existing chemicals, because I really think one of the greatest opportunities is in the new chemicals area. And if you look here, what I've presented is just a simple stage gate process. And for those of you who may or may not know, but all industry practically uses this stage gate concept to commercialize a new product. And NAMs has some real motivation in this process, because up until then, you can see by this red box here under commercialization, we really never got to assess the safety profiles of our new chemistries until we were well down the road of product development, which means we had invested a lot of time, a lot of money, and a lot of effort. And if we had something that was unfavorable, we lost a lot of investment. So NAMs gives us the opportunity to really start to look at health and safety profiles in materials that we haven't even synthesized a gram of chemical yet. And so many companies have recognized this and have begun to use this and have begun to work very closely now with their R&D facilities to say, as you're making design choices to, make a better product for performance, there's also health and safety effects that could go along with that. And how do we now build that into the stage gate process and make that part of our decision-making tool for what we will advance further on into product development? And so, we're generating data in these NAMs already. Many companies are using them as they look at new product development. And the benefit is that, they're quicker, obviously, and they're less costly. And so that allows us to get materials to market quicker and have greater confidence that when they get there, that they're going to be a more favorable commercial product from a human health and environmental perspective. So, that's really the motivating factor, I think, for companies. So my company is a very small company. We have 350 employees, but we have a tremendously active R&D pipeline where we're using these tools already as we're looking at launching new products. And so I think that anything that we can do from a perspective to start to understand and partner with industry on how they're using these in the very early stages of product development and how that data can actually begin to feed into the regulatory process will be important. So I think that, as we look at how we're feeding the NAMs into the safety evaluations, the companies are going to have data on many of these NAMs, and is there a way to bring that in as we're looking at nominating NAMs to be considered? Because this isn't going to be something where a company will develop a NAM. Quite frankly, we'll partner with a developer like Patrick's organization to test these things on our chemicals and generate that information. So we would be partners in submitting NAMs, and there would be a motivation for us to want to submit in partnership certain NAMs that we think are going to help us do these early safety assessments. So I would ask, Steve and Gino, as you're thinking about this, I know you're thinking of it with the end in mind. How am I going to use that data in a TSCA PMN or an existing chemical? But I guess I look at it as kind of a continuum. We're building our information on health and safety of those materials as we go along the R&D process. And is there a mechanism as well to not just get a NAM certified, but to maybe this is where the IATA piece comes in, but looking at these additional pieces of data that might be generated that might support a weight of evidence assessment, although they may not be a formal NAM evaluation. And so that might speak to looking at this in the context of an IATA where not everything has been validated or fit for purpose, but when we pull it together and look at it, it would be more of a weight of evidence approach. But I think that these have some tremendous value. They fit with the goals that I think EPA has to move away from, the reliance on animal testing, and with Warren, what you've expressed as far as getting safer chemistries on the market. And I do think that industry will want to, be part of helping to submit these NAMs. But I think we have to make sure that we're not just looking at the NAM itself, but the experience that went behind it as we were generating that data. And then, Steve, you raised an interesting question about, should NAMs include things like predictive methods for phys chem and those types of things. I'm going to give you my opinion on that. I mean, I think we've had predictive models for a long time, so we've been using those for phys chem for a very long time. I don't think they're the same as NAMs. I think NAMs comes with a special context around truly trying to move away from our reliance on animal testing, and that makes them unique. And I think if we start to dilute that definition, it might draw focus away from what EPA is really trying to do as they're trying to reduce their reliance on animal testing. So I'm kind of in the camp where I feel like, NAMs has that specific goal that we're really trying to move away from our reliance on animal tests, become more human relevant models. And these other predictive tools we've used for decades, I'm not so sure that they really fit with that goal. So with that, I would just say, ideally as a company, we have a huge motivation to want to use NAMs. And we would like to see that these NAMs that we're using early in product development would somehow build and fit into the final regulatory consideration and the safety assessment.
Great, thanks
and
so much.
With that, I'll close.
Perfect. Then we will move on now to our fourth and final panelist for today, Kristie Sullivan. Kristie is the Vice President for Research Policy at the Physicians Committee for Responsible Medicine, where she leads efforts to replace animals with more human-relevant methods in research, education, and testing. She is a founding member of the American Society for Cellular and Computational Toxicology and has been involved in efforts to develop and implement non-animal approaches at the international and federal level. Kristie?
Thanks, Amy, and thanks to all of you for listening, and thanks in particular to Steve and Gino for pulling together this event, because I think it's really a nice way to get some feedback and to kind of advance some of these questions that honestly many of us have been thinking about for a long time. So if you could just go to the next slide. And there really is a need for this pathway to suggest NAMs to be added to the TSCA 4H list. And so I really appreciate EPA thinking about how to develop this process and putting it together. So the list already includes methods that have been adopted by, OECD or ICFAM or EPA. And so I think what we're talking about here are NAMs nominated via this form that are likely not to have undergone a traditional what we think of as a sort of validation process with a lot of reference chemicals and interlaboratory and intralaboratory validation. And so I think what I heard from Steve and what I've also heard from some of the panelists is that there's a need for some flexibility for different types of NAMs and decision needs, that the process should focus on whether the NAM fulfills the information needs for a context of use. And I think the process provides some reasoned guidelines for assessment of relevance and reliability, depending on the NAM and its envisioned use. I think we want to avoid, and I'm not saying that this was suggested, but we want to avoid sort of like people feeling like they have to go through a years-long validation study before being comfortable submitting through this process. And so I do appreciate kind of the intention to ensure the process offers flexibility, and that there's focus on demonstrating the NAM is reliable and relevant for that envisioned context of use. Because I'm a sort of a rule follower, I decided to base my presentation on some of the questions that Steve was posing earlier. So what would motivate me personally to submit an NAM? And I think coming from my perspective as an organization that advocates for the use of more NAMs, I would want it to be used, kind of building off of what Pam said. Use of NAMs in early development is by companies is really well known. We've known about this for a long time, and I'm hoping that this nomination process can help companies and stakeholders share these strategies so that they can be used by EPA and other companies more widely. Just some thoughts that came up as I was thinking through this question from a submitter's perspective. Nominators might not have all the information required for the application. Is that okay? Or should they try to get in contact with the developer and make sure that they have that information? What does EPA envision by an independent review? Is a submitter expected to convene a peer review, or what are some of the thoughts behind those words exactly? And what represents good coverage of toxic chemical space? There are lots of different kinds of chemicals in the toxic chemical space. There's lots of different uses of chemicals. Does it mean a certain proportion of the TSCA inventory? A NAM may have a very specific application, which would still have an impact on animal use and utility for a certain chemical class or category. And we want to be open to those solutions rather than wait for something that covers everything. From EPA's perspective, so what information on the form is nice to know versus really need to know? Is there some thinking behind that? And EPA might know better than the submitter how a NAM can be used to meet EPA's needs. And so that leads me to think maybe there needs to be a little bit of back and forth to see exactly how EPA might envisions it would use a NAM. So the next slide, please. If an integrated method is submitted, do we need to evaluate the constituents independently? This is a really compelling question that we're kind of wrestling with right now, right? And I think individual methods may have specific technical limitations or training sets that define something like a domain of applicability. But beyond that, I don't think we necessarily need to evaluate all the components individually, because that is the purpose of an IATA or a defined approach. They're meant to work as a whole, including the decision procedure. And so it almost wouldn't be fair to kind of judge it by the kind of evaluation of all the components. And then just a couple of thoughts about evaluation of NAMs. This term domain of applicability has a really specific meaning and implication for QSAR models, which are built from a training set of specific types of chemicals. In vitro methods aren't necessarily or shouldn't be bound by this really specific set of test chemicals unless there is an observed technical limitation. And the relevance of the NAM should be considered within the context of use. So we should be careful not to discount NAMs that don't show exact fidelity to animal tests that we currently use for a certain endpoint. And I really support the idea of bringing in the concept of the adverse outcome pathway as a companion to evaluating a NAM by supporting the biological or mechanistic relevance. Next slide, please. These last couple of questions are really good, but slightly different. I would say actually, yes, this CAM information can feed into IATAs and so could be considered. I know they're not necessarily new, but I think they fit within that concept of informing which in vitro or in silico testing approaches are most relevant. That information can also facilitate exposure-based waiving. So it really depends on what is the context of the decision you're trying to make. And I also just wanted to briefly mention partial replacement. So a NAM can offer a way to replace an animal test for a specific chemical or a specific mechanistic space. And it brings to my mind some of the other approaches and strategies that could be used to add to the overall information that contributes to a regulatory determination, but isn't going to give you a quantitative risk assessment, something like assessment of metabolites, comparing toxicity of different formulations, or read across threshold of toxicological concern, all these pieces of information that lead to an overall evaluation, and just making sure that is the form flexible enough to accommodate these types of NAMs. And then the last slide, please. I just want to end with kind of a few final considerations, making sure that what we heard today is reflected in the form. Do readers of the form interpret it as having context and NAM dependent modules that can be filled out as relevant or put to the side if not? I think it would be a good idea to try to consider testing the feasibility of the form and the nomination process with different types of NAMs. Perhaps we can make a series of case studies exploring with potential nominators what it's like to fill out the form and go through the process. And then how is EPA planning to handle the nominations? Are they going to be posted online, EPA, or are they going to comment on them? Curious about that. And I just think, just echoing what Lauren said earlier, this nomination process is a really key feature of the agency's strategy, and so I really appreciate it being sought, but it's sort of the first step, right? Where there's going to need to be a lot of education, outreach, and training to make sure that companies are following the process, that they're using the NAMs list. We are looking at ChemView and finding that even after the list has come out, companies are submitting data, in vivo data, where there are methods on the list and that could replace those methods. So I think this is a great first step, and I'm looking forward to the rest of the discussion and continuing to work on it. Thanks.
Thank you, Kristie. And again, thanks to all of our speakers for today. We now have time for anyone on the line to ask their questions or provide feedback for Steve. And to do this, again, we're using the questions section of your GoToWebinar toolbar. You can type it in and we'll read out the question for you. And while those questions are coming in, I wanted to alert everyone to 2 upcoming meetings of potential interest. Information about these meetings is on the screen now, and we'll also paste the registration links into the GoToWebinar chat box. So if you're interested, you can sign up for those if you haven't already. And Steve, questions are coming in. Before I get to those, is there anything that you wanted to say now in response to the questions and comments from our 4 panelists?
No, not in probably 20 minutes. I would need like a whole nother 30 minutes to address those. That was a lot of information from 4 panelists in about 20 minutes. But I do appreciate the comments. I do find it interesting. Just I'll comment on one thing, even though it's probably not the most important thing, is that this sort of tension that exists between, the sort of difference of opinion that exists about whether or not a NAM necessarily has to replace an animal test. We saw one view reflected by Pam, where she, I think, if I'm restating her position correctly, that thinks that NAM should replace an animal test because that's sort of the purpose of this effort, sort of the raison d'etre for the whole thing, versus the view that was articulated better than me by Kristie, that based on, we use a lot of phys-chem properties in TSCA decisions already, and some of those decisions, like for instance, a phys-chem decision or phys-chem based NAM might be used to make a prediction about exposure that then would preclude an animal test, even where we might think a hazard might exist because exposure is not really a possibility. So there are real world consequences using phys-chem methods vis-a-vis animal testing, but this sort of tension about where a NAM sort of fits in the middle of that, and how whether or not something that doesn't necessarily replace an animal test should be called a NAM, I find interesting. And I can just say internally we have people on the TSCA NAM team who hold both positions. Both positions are well represented on a TSCA NAM team and are strongly held by different people. So.
Okay, thanks, Steve. Then I will jump into the questions and comments that have come through. There were a couple along the same themes. I'm going to try to group some of these into one question. One is about reproducibility between lab to lab, and asking about non-testing approaches such as QSAR, read across, categorization, and they also specifically mention EcoSAR and EpiSuite.
Yeah, so I don't really know that particular sort of sub-element of relevance is going to be particularly useful for something like a QSAR model, right? So if a QSAR model is, if you get 2 QSAR models and they're basically using the exact same method on the exact same set of descriptors with the exact same training and test set, then my guess is, and I'm not super well versed in QSAR models, but my guess is they should come up with the exact same model. That some of these subpoints are really included. They're not necessarily, there's not necessarily overlap for every NAM type with every single one of those sort of requests for information, and so I don't know that one's particularly relevant for QSAR so much. Like I said, I think if 2 people use the exact same set of descriptors and the exact same method, they should come up with the same model, more or less. So I hope that answers that question.
Yep, thanks. Let's see, the next question is, does the nomination also depend on the type of chemical? For example, what about mixtures testing?
My hot take on that would be no. If you've tested mixtures, personally, I think that would be great, because it would show that NAM could then be used to test mixtures. Mixtures is one of those sort of tricky chemical categories in TSCA. I want to say that I'll throw out a number. I think it's pretty close to right. If Gino's still on, he can correct me, but I think on the TSCA inventory, it's something like 20% of the things that are listed on that chemical, might even be higher than that, are defined or undefined mixtures. So if you look at mixtures as sort of a separate chemical category, I mean, every mixture is obviously made up of constituent individual chemicals. It's one of those problem categories. So no, I would look at it as, personally, I would look at it as a plus if your NAM could handle mixtures.
Great, okay.
And this is Gino,
go ahead, Gino.
No, I was just gonna say, I don't know what the number is, Steve. I just wanted to let you know I am still here.
That's very reassuring.
Yes, we're glad you're still with us, Gina. All right, then let's see. Next question. I recognize that the initial accepted NAMs list was based on many of the accepted OECD methods. As new methods become accepted by OECD, will they automatically be included on the TSCA list of accepted NAMs?
Yes, again, I'll give Gino the opportunity to comment because he was definitely more involved with the construction of the current list. But my understanding is, as long as it's something that's relevant for TSCA, my assumption is that if it's gone through the OECD process, then it more than meets the requirements for use in TSCA.
That's correct, and yes, that's a good assumption. If it's on the OECD list, it'll be on our list.
Excellent. That's good to hear. Okay, then next up, you've discussed transparency as a feature of an ideal NAM. EPA is currently implementing its new strengthening transparency policy. Can you discuss how this policy might affect NAM acceptance?
I would like to. I don't. I mean, Gino can weigh in on this one.
Yeah, unfortunately, I think that is a TSCA specific question, and that CBI transparency effort is done by a separate group of people. That's a good question that I can look into, but I don't have an answer for now.
Okay. Yep, that sounds great, Gina. Thank you. All right. So there's a question here about how long the registration of a NAM takes. So I guess that's a question about going through this nomination form and about how long it might take from when it's submitted until when it would be added to the list.
That's a very good question. I wish I had a more satisfactory answer right now. The nomination is really just the very first step, as Kristie alluded to, and I think her last slide. There's a process that still has to be fleshed out. We still, besides the actual nomination form, I mean, to me, the very next step then, again, and some of this is, you guys watching us sort of make sausage internally here at the EPA, and I know it's sort of mundane, it might be a little mundane and boring, but we have to basically establish a vehicle for submission. Is it going to be emailed to an email address? Is there going to be a web form that people fill out? Who do the web form go to? Is there somebody who essentially sort of triages the nominations and then it goes before, I'm assuming, the TSCA NAM team, but that hasn't necessarily been settled yet. How often are they going to convene and take up nominations? What does the evaluation process look like? What's the process for the NAM team to get feedback to nominators and or developers for more information if those are required for evaluation? And then ultimately, what's the method and vehicle for the decision process? And is all that posted on a website? Is it public? These are things that still all have to be fleshed out. So I guess to answer the question is, that timeline would depend on what that process looks like. I mean, I would hope that it could be relatively quick for nominations where we do not need additional information or feedback. So.
Okay, and then there's a question here which I think's come up a little bit, but I'll ask it again just to make sure it's clear. Can NAMs be used to completely replace animal studies, or will animal studies always be required before launching a product?
I'd like to get Gino's take on this. I think that it probably depends on which animal study you're talking about. There are some animal studies, I'll just throw one out there, and this isn't company policy, this is Steve Simmons' personal view. I mean, would it be possible to come up with a integrated approach of NAMs that could eventually replace the 2-year carcinogenesis bioassay? I would like to think so, I hope so. Whereas, is there a IATA that's going to replace a multi-gen repro study? That I'm a little less sanguine about the possibility of that. So I really think it depends on which animal test we're talking about replacing. And then, how ultimately it comes down to how much of the animal test can essentially be distilled down to sort of reductionist puzzle pieces that can then be, you know, assayed independently and then reassembled to look like an animal test.
This is Gino. Can I add to that? And thanks, Stephen. Thanks for the question. I think in the TSCA world, where it's a little different than like the pesticide or drug world, where there's no minimum data set, where there's no data requirement for toxicity. So that means we use whatever is at our disposal, and then we would make some requests for animal testing based on what we think is missing and important to making a risk decision. So given all that, the way Section 4 is written, reduction of testing in invertebrates, I mean, we importantly need to go through all these steps before we can ask for an animal test if we think it's necessary. So from a practical standpoint, I guess we could if we think everything's exhausted. And as Steve alluded to, if there's an endpoint that for which there's nothing currently available, be it an IATA or a single NAM, we might go down that road. But you know, this is a journey, and so I do, and so anyway, I think that's an important distinction. So we don't have a defined data set, so we can't like waive depending on exposure pathways or considerations or available other information that might be submitted with a chemical. It's a different way to look at it. So, but this is an important beginning in that journey as we try to figure out the best way to reduce testing invertebrates.
Thanks, Gino. And we have a few more questions here, and there's actually one for Pam. Pam, if you're still with us, well, I'll ask you one, and before going back to a couple more for Steve and Gino. So Pam, it's okay, good. The question is, it's great to hear that companies like ANGUS are making such progress in applying NAM-based design strategies. In the past, industry may have been hesitant to invest in such upfront testing. Do you see that trend changing, or is your company unique in its approach?
Oh, no, we're not unique at all. Again, you know, we were a victim of the limitations of the animal test, right? As I said, if you think about how product development works, you have to have some level of test compound to test in animals. And in many cases, the animal studies consume large amounts of test material. And so it was really nearly impossible to do any animal testing, or I mean, any early safety screening because of that constraint. So NAMS has really given industry now the opportunity to do these very early assessments. You know, these in vitro tests take very, very small amounts that a chemist can synthesize at a bench, and we can start to help, you know, our R&D make decisions on which materials look better to advance in product development. So.
Great, that's great, Pam. Thanks for that. And I agree with you. In my interactions with industry, I've been seeing a lot of the same, a lot of interest, which is fantastic. Okay, a couple more questions, Steve and Gino. One that Patrick sent in regarding the third question about the need for a NAM to replace an animal test. I'm a little confused about the framing. As you know, TSCA encourages the use of a tiered process. I interpret that to mean that we will be using faster, cheaper NAMs to inform the downstream decision making and drive requests for additional information. Most TSCA decisions don't necessarily depend directly on an animal test using the test article, but most could benefit from the incorporation of NAM data into decision making. There wasn't exactly a question, but it was I think there was a question in there.
Yeah, I'll let Gino take a whack at it, but I mean, I'd say I agree with Patrick. I mean, certainly with existing chemicals, I think that's definitely the case. I mean, I know, as Gino said earlier, especially with new chemicals, you know, a lot of times there's very little data, if any. So again, whether or not it needs to, replace an animal test, I think Patrick points to the promise that, NAMs can be used a lot more widely than animal tests are currently being used, or at least to cover much more of the chemical space. And so, as Gino was saying earlier, that provides data for chemicals for which we currently don't have any animal or otherwise.
Yeah, thanks, Steve. And I don't know, Patrick, when I went through my earlier description, I was hoping that might address some of this, but I think specifically about the tiering, I mean, in the new chemicals program, we are starting, we've always done that in different ways. I mean, the simplest example in terms of doing an animal test, we might ask for a short-term one before a longer-term one. But I think the more NAM-related ones is when we, and I think our colleague Todd Stediford and others are in the process of publishing some work on lung categories and the use of in chemical tests to help inform whether there is a need to do some animal testing from, when we're worried about inhalation exposures. And, like two simple examples would be a surface tension test. If we're worried about surfactancy or waterproofing, and the idea that chemical properties measurement will go a long way towards determining whether or not we need to be concerned about that endpoint if the chemical, if there's inhalation exposure to that chemical in the workplace. And then the other thing was, if you have a something that you're worried about, and most of the time, as Steve alluded to in the PMN process, we talk about PChem properties. And when we're thinking about evaluation, and we talk about water solubility and log KOW, the optional water solubility, one of the more important things for inhalation is the idea of biosolubility. So you have a simulated fluid in the lung fluid that is the more appropriate measure of concern about exposure in the lung and how it may or may not be cleared based on solubility in that simulated fluid. So we do think about those things. And, there's a number of our folks that are doing that kind of thinking. So I hope that helps.
Thanks, Gino. And we just have a few minutes left. I'm going to ask one last question and then we'll wrap up. Steve, how will accepted best practices for lab methods be considered and integrated into nominated NAMs? For example, cell culture best practices, technical replicates used, etc.
Yeah, that's actually a very good question. And to be honest, one I haven't really thought about up till now, but it's an important consideration, right? So, we sort of touch on this very superficially when we ask about quality assurance in terms of acceptance and rejectance criteria. But yeah, I could see where there might actually be, maybe another question or two that we need to probably add to the form to address, GLP, particularly when it comes to things like cell culture and things like that. That's actually a really, really good point.
Okay, Gino, any final thoughts before we wrap up?
I just want to thank Steve for presenting this and the panelists for presenting really great feedback, and the people listening, they were obviously engaged and provided good questions. So I appreciate that.
Likewise, I echo that. Thanks, Gino. And just a reminder that this recording and Steve's slides will be posted on our website within a couple days. And this is an ongoing series, so please keep an eye out at that same webpage for future webinars. Enjoy the rest of your day. Bye.
Thank you.
Bye. Thank you.
Bye.